So then I will start. Welcome, everybody. At first, I will introduce myself, I think, shortly. I'm not a biotech, sorry. I'm not, sorry. I have here always on my screen, Abdelbary, this meeting will be recorded and I cannot close this picture. Abdelbary?
So you cannot.
Uh,
Okay. So what you wanna me do?
No, I don't know if you can do it because it is now when you started with it is in the mid of my screen. Let me see if I can close it. Yes, now sorry, I will start. So
Yeah. So you can.
mm, okay,
So
okay.
you.
Now I can start. So
Yeah.
now, I would like to give a short introduction about my person because I'm not a biotechnologist like most of you. I'm a vet and I'm using flow cytometry about 25 years now for the evaluation of sperm quality, but more under clinical aspects. But we will see. I think most of you know that since 350 years about now, sperm can be examined because we have the microscope. And until I would say about 40 years ago, this was the main thing, or nowadays it's still done with the microscope, but nowadays more and more we have other systems like the computer-assisted sperm analysis system, or now the flow cytometry to determine sperm quality. And there's a tremendous increase of using this technique. Flow cytometry was originally mostly used by immunologists and people working in other clinical fields, but since about 25 years, there is a tremendous increase using system in spermatology. You see here about 50 papers per year in 2000 about, and now we have 250 paper if we are looking on Scopus and the number of publications by using this system. In my presentation, I would like to give you at first some information about the basics of flow cytometry, because I think there are also veterinarians or first-year students. Maybe for them this technology is new. Then I would like to show you why we are using flow cytometry instead of the conventional methods like a microscopy for sperm analysis. Then I would like to show you some examples of flow cytometric assays studies we have done with this technique, and but last but not least, coming to the conclusion. Starting with the basics of flow cytometry, we need normally for flow cytometry different systems. At first, we need a laser with the emission of light. Most often, the 488 nanometer laser is used, but there are also UV lasers and 630 nanometer lasers. Nowadays, most of the machines have 3 to 4 lasers. And we need a fluidic systems where they, for example, the cells are going through, and also the sperm cells, and then they are excited by the laser. The sperm, you have to do the staining before you give it through the fluidic systems. And then there's the optical system with detectors. And then you can measure the emission of the fluorescence of these cells. And these signals have then to be converted from analog to a digital signal by using an electronic system. Here's another view of the setup for a flow cytometric system. It's a little bit more complicated. You see here's a laser, here's a fluidic system, and you have a forward scatter to measure the size of these cells, and here a side scatter to measure the granularity. And we have these different fluorescence mirrors and detectors to measure here the fluorescence intensity of the different colors. And now coming to the advantages of flow cytometric assays compared to conventional methods for sperm analysis. Very important is that you have a very objective and quantifiable results. You can measure the fluorescence, not to say like the eye, it's more or less green. You can measure the fluorescence intensity. For example, you see here two populations, one population with a low intensity and another population with a high intensity, fluorescent intensity. And it is possible with this machine to measure a lot of cells in a very short time. Normally, if we are measuring, for example, sperm cells, we are measuring 500 sperm per second. And if we examine one sample, then we are measuring the fluorescent intensity in 10,000 sperm. And it takes about half a minute and not 10 minutes like if you are doing microscopy. And if you see here the accuracy of a method in relation to the number of analyzed cells, here sperm cells, then you see here the accuracy quantified by the coefficient of variation that you see if you measure, for example, 300 cells, like in you doing microscopy, then the coefficient of variation is 12%. But if you're doing it with 10,000, this variability is only 2%. So there's a tremendous increase in the accuracy of the measurement. And another good advantage of this method, the most important advantage, that we have a lot of different fluorescence probes to examine the different characteristics of sperm. At first, we can measure the number of the sperm cells and the concentration. And the most important thing is to distinguish between live and dead sperm. And then we have here, for example, a lot of different probes to examine the viability of the sperm. Then we can characterize the destabilization of the plasma membrane of sperm. For example, this is very important in sperm during the capacitation or to examine the damage because of the cryopreservation of the sperm. Then we can measure the oxidative stress of the cells, the mitochondrial status of the cells, the chromatin status, the calcium uptake, the protein phosphorylation, and other surface of cytosolic biomarkers. We have ion probes and the pH value. So these are only some examples. And then you have, for example, if you're measuring the motility and the morphology, you have only two characteristics of the sperm. And here you have about 10 different characteristics of the sperm cells. And now to show you some examples of flow cytometric assays we are routinely using. This is, for example, as I already mentioned, the most important point is to measure the plasma membrane integrity of the sperm, if the sperm are viable or not. And here, most often there is used cyber-14 in combination with propidium iodide. And these are two stainings. They are showing the DNA. And the difference between the cyber-14 and the PI is that cyber-14 permeates only the plasma membrane if it is intact. So, sorry, it can go through the cell in both cases. And propidium iodide is only able to penetrate the cell if it is damaged, the plasma membrane intact. And therefore, you see the red color if the sperm cells or the plasma membrane is defect, and green if it is intact. And if you look on a dot plot here, and this is very typical for flow cytometric image, you see here a population with more green fluorescence. These are the plasma membrane intact sperm. And you see here a population with more red cells, and these are plasma membrane defect sperm, damaged sperm. And here is debris, for example, these are particles, egg yolk particles, for example, in the sample. And you are only measuring here the percentage of the green, the population with the green fluorescence and the red. And then you can say, for example, here are 60% show more green fluorescence, and these are the sperm with an intact plasma membrane, and red, here are the sperm with a defect plasma membrane. And another important parameter, the expert on this field is Abdelbary, is the mitochondrial membrane potential. Also we examine the activity of the mitochondria, very important, for example, for the motility of the sperm. And we are using here JC1. JC1 shows a green fluorescence if there are in monomers. If they are aggregated, then they show an orange fluorescence. And the interesting point is that in intact mitochondria, the membrane potential, the negative membrane potential is very high with minus 180 millivolt. And if there is only a low in normal cells, not in the mitochondria, it's about 60 millivolt. And then you have here aggregate monomers with a low membrane potential, and with a high membrane potential, you have orange fluorescence. It means that if the sperm show a high mitochondrial membrane potential, they're showing
بتكلم كلام مهم، انا حاسه كده. بيقول لك من بوتنشال بيشرح حاجه.
an orange fluorescence, and if they are in monomers, they show a green fluorescence. And this is shown here. Here you see the mid-piece of the sperm, and with more orange fluorescence. This means this sperm show a high mitochondrial membrane potential. And here you see more a green fluorescence. This sperm has only a mitochondria with a low mitochondrial membrane potential. And another point which is important for the fertility of sperm is the agrosome. The agrosome is normally binding then to the oocytes during the fertilization. And we are looking at the agrosome by using peanut agglutinin in combined with FITs, which is showing a green fluorescence. And if there is, for example, a disturbance of the agrosome, then there is a green fluorescence. This means here fits PNA positive. You see here, red is the PI of this because it was combined with providium. This means the sperm is dead. And here you can see the green fluorescence of the agrosome. Here this sperm cell does not show a green fluorescence. It could be that there is no agrosome or there is no binding because there is no damage of the plasma membrane. A very important parameter is the examination of the chromatin structure. We are using here the sperm chromatin structure assay. This was developed about 40 years ago. It was published by Evenson in Science at this time. And this is a very important parameter. They are also using it very often in human medicine to detect subfertile men with a high DNA damage. At first, when we are doing this staining, we are using hypochlorite acid to denaturate sperm DNA. And this stress causes that some of the sperm who are sensitive, they show then a single-stranded DNA. The stable sperms, they still show a double-stranded DNA. And we are using for the staining acridine orange. And this is a metachromatic staining. It means if the acridine orange is binding to the DNA, which is double-stranded, this is the intact DNA, then it shows a green fluorescence. If it is binding to single-stranded DNA, this means damaged DNA, then it shows a red fluorescence. And if you are looking at the microscope, then you see here on the left side sperm with a damaged DNA showing the red fluorescence and sperm with a green staining. This is with double-stranded DNA with an intact DNA. And as I already told you, with flow cytometry, we not only can distinguish between red and green, we can quantify this fluorescence intensity. And normally, so we are measuring the DNA fragmentation index, is DFI. And this is, DFI is measured in each sperm cell. And it is calculated by using the red fluorescence divided by the total fluorescence, means red and green fluorescence. And if we are then looking at the flow cytometric image, normally we see when we are looking at the dot plot, here are the sperm with a high red fluorescence, and these are the sperm with a damaged DNA. And this is a population with more green fluorescence. These are the sperm with intact DNA. And here's again the debris. And because here the distinction is not so easy, then therefore we are using a histogram. And if we have this histogram showing here this DFI value, the percentage of red fluorescent or the quotient red fluorescent divided by green and red, then you see here one population with red sperm and one population with green sperm. And this population, this is the sperm with a high DFI value. And normally this percentage of sperm is quantified. And to show you an example, you see here a dot plot from a stallion with an intact, with a relatively low percentage of sperm with a damaged DNA, more intact DNA. And here is a stallion with a very high percentage of a damaged DNA, the dead cloud, compared to the green cloud. You see it here on the histogram. On the right side, here is also damaged sperm with a damaged DNA. And here, again, here you see the population is much higher. You can also have a look under the microscope. You see here only one orange sperm. And here there are a lot of sperm with an red or orange color. Nowadays, we are using multicolor flow cytometric analysis for the examination of sperm. We have published one of these assays we developed in our lab and related it with the bull fertility. Why we most often examining bull fertility as a model compared to stallion fertility is because bulls have a very high amount of offspring in contrast to stallions. So bulls often have thousands of, sometimes hundreds thousands of offspring, and stallion often 100 or 200. So the accuracy of the pregnancy, of the determination of fertility is not so high in stallions like in bulls. For example, bull assays are, for example, even for human medicine, the bull is used as an animal model to determine or to examine the reliability of the assays. And here we developed a multicolor assay. It means nowadays, as I already mentioned, our flow cytometers have the possibility to have different lasers. Like this flow cytometer has, for example, a violet laser, a blue laser, and a red laser. And we have then different emissions if we are using different probes. And then we are measuring different characteristics in sperm. In sperm, the metabolism is important. Therefore, we measured here the esterase activity in the sperm. Very important is the calcium level in sperm. It's shown in several studies that sperm with a high calcium level are subfertile. We are measuring the acrosomal status, I already mentioned, the plasma membrane integrity, and the mitochondrial membrane activity. And then we combine this in one assay. And the advantage is that, for example, then we know then the percentage of sperm who show a high esterase activity, at the same time low calcium level, no acrosomal damage, no plasma membrane damage, and a high mitochondrial activity, to show all these characteristics in one assay, and not to say one single characteristic in one assay. So for example, then if we are doing only one assay, then or with one staining, it could be that there are mitochondria with a sperm with a high mitochondrial activity, but a low calcium level. And but we don't see it by using only single assays. And now coming to some examples of our studies. At first, when we started with the assays, we controlled the reproducibility of the assays, and we measured the intra-class correlation coefficient. And this was very high, more than 88%, up to 98%. So I also controlling always, for example, my graduate students before they are starting with their trials to control also the reproducibility of the technique of the student. Then we compared, for example, the percentage of plasma membrane intact sperm with the mitochondrial membrane potential. There was a high correlation in this study, but I also have to mention that there sometimes, not in a lot of stallions, but in some stallions, also in humans and in other species, it could be that sperm show an intact plasma membrane, but they show a low mitochondrial membrane potential. And we have done a trial and induced heat stress in bulls by using scrotal insulation to see what's happening after heat stress on the testicles. And here we used 3 different assays: the plasma membrane integrity with the CyberPI, the mitochondrial membrane potential with the JC1 assay, and the acrosome intact. We measured the acrosome intact sperm by using the FITS-PNA. And you can see immediately, or 1 week after the scrotal heat stress, there was a tremendous decrease of the mitochondrial integrity and the plasma membrane integrity, and an increase of the acrosomal damage. It took about 6 weeks after the heat stress until the sperm got values like before the study. Even more interesting was the effect on the DNA of the sperm. And here you can see after the induction of heat stress that there was a tremendous increase of the percentage of sperm with a high DNA fragmentation index. It was more than 55%, but this increase was not as was a little bit later, the maximum values compared to the values with the plasma membrane integrity. But what was more interesting was that if we are looking on the morphology of the sperm, only half of the sperm, or 25% of the sperm showed morphological anomalies. It means that you cannot detect the sperm with all the sperm with a DNA damage by using morphology. This is important because a lot of people, they think that if you are measuring the morphology of the sperm, you also detect the sperm with a DNA damage. It's very important, for example, for human medicine, because in human medicine, they often use morphology to decide which sperm cells they are using then for doing intracytoplasmatic sperm injection. And it could be that they inject, for example, then sperm with a DNA damage because they don't realize it by using this method. And here you can see again the relationship between head abnormalities and the DFI value, or the percentage of sperm with a high DFI value. So you see there is some relationship, but there is not a high relationship. In another study, we examined the seasonal variability of the DNA damage in stallion sperm. This study was done in Germany. And you see here the percentage of sperm with a DNA damage. It was lower in December compared to March and July. Not surprising because the temperatures are lower, but for us it was surprising that it was already increases DNA damage in March. And in March, there is no heat stress in Europe. For example, today we have snow here in April. And it means that there is not only heat stress, there are also other factors, other stress factors leading to this increase in DNA damage. And if we examined in this study 9 different stallions, and if you are looking on their values in December, March, and July, and look at the different stallions, then you see this stallion number 1 showing an increase, like we accepted. This stallion number 2, he had high values on in March. There was something happening with this stallion. The people on the stud did not realize it. Here, for example, stallion number 4, he has a tremendous increase in July like we expected. But we had, for example, also a stallion who improved between December and July, and also number 8. So it is important, we recommend to the people who are collecting semen and produce cryopreserved semen to control regularly this DNA damage of the sperm of the stallions, because this DNA damage has an effect on the fertility of these stallions. Here you see the pregnancy rate per season, and here stallions, these were 4 stallions with a high DFI value, showed a distinct lower pregnancy rate per season compared to the others. And this effect on fertility was nearly shown in all species, means in human, in pigs, in bulls, in stallions, and in dogs. So this is a very important assay for determining the fertility of animals. Another study we have done, more detailed study, we examined here 15 stallions every month by using this DNA fragmentation, and we could show it, again that there are seasonal changes. But if you see here the points here, this means these are values from different sperm samples at 0 hours, 24 hours, and 48 hours, then you see the variability between animals was very high here in stallions, but we can see this also in bulls. So here you see 50 different bulls, and you see the plasma membrane and acrosome intact sperm. Here is not the DNA damage, and you see here a very high variability between the bulls. And what is even more important, the variability within the bulls. It means that, for example, if you are doing variance component estimates, that 50% of the variability is depending on the bull, and 50% within bulls, and 50% between bulls. Therefore, we are examining from the AI station each ejaculate. So we are doing, for example, for one AI stations, we are examining 5,000 sperm samples per year. And here, for example, shows you the results from last week I got from this quality control. And we controlled, for example, here from this AI station in the fourth quarter of 2023. And the first quarter, sorry, it should be 2024, it's a mistake. And you see what we are seeing here, this is the frequency distribution of the plasma membrane integrity of the sperm. And you see that normally we have here this cut-off value of 40%. If the samples show less than 40% plasma membrane intact sperm, we don't use it for AI. And we had here only 2% of the ejaculates could not be used because they were under the threshold. It was the same in the last quarter of last year and the first quarter of this year. But what we have seen also, if we are looking every week on the sperm quality of the sample, we see here a tremendous decrease of the plasma membrane integrity of the sperm on the AI station in between weeks 11 and 13. It means there is something happening on the AI station, and we have now to find out why we have this decrease in the sperm quality. And another study I would like to show you is, for me, it was one of my most important studies because at this time we examined the effect of the prolongation of the equilibration time for several days and its effect on quality of cryopreserved bovine sperm. You have to know up to this time, most of the AI stations, they were using incubation times of about 6 hours. Nowadays, I think worldwide, most AI stations, they are using now our protocols with incubation time or equilibration time before freezing of 24 hours up to 72 hours because of the results of these studies. Why we have done this is because our AI station, we are collaborating now since about 20 years. They thought, oh, we always on Friday, when we collected the sperm, we have a very low sperm quality because we are doing the cryopreservation on Friday a little bit earlier. And I asked them to wait for a longer time. And before we started, we did an experiment. So we used 8 bulls and with different age, using CASA system to examine the motility and flow cytometry. And we did 2 trials. At first, we examined the effect of the prolongation of the equilibration time up to 4 days on the sperm quality before and after cryopreservation. And in the field trials and on the AI station, we looked at the effect of the increase of the equilibration time on the AI station, on the sperm quality. So motility, we also compared different extenders. Triladyl extender is the conventional extender with egg yolk particles. Andromed is extender with soybean instead of egg yolk. And optic cell is a new extender also without egg yolk particles. And you see here the percentage of rapid sperm. And you see if we increase the equilibration time until 48 hours, there was no effect on the plasma membrane integrity in most of the extender. But if we used the Andromed extender and waited for 72 hours, there was a decrease of the percentage of rabbit sperm in contrast to the trilateral and optic cell extender before the cryopreservation. Here you see the effect on the plasma membrane integrity also before the cryo is similar to the others. And what we have seen here is that there was a tremendous negative effect on the DNA damage if we used the trilateral extender. It was not possible to wait too long if we're using this trilateral extender. Then we had a high increase of DNA damage in contrast to the other extended without egg yolk particles. We examined then on the field the proportion of samples with a plasma membrane integrity and acrosomal integrity. And you see here in 2012, this was the year before we started with the longer equilibration time. There were about 20% of the sperm samples did not fulfill the criteria of 40% plasma membrane intact sperm. And in 2013, when we started with the longer equilibration time, then we had 5.5% of the sperm with a lower percentage of plasma membrane intact sperm, less than 40%. So we have to throw away at this time only 5.5% of this sample compared to 19.4, means about 15% less. And if you think, for example, what are the costs for the production of one semen sample, it's, let's say, about €1. So, and they are producing 5,000, hundreds of thousands of samples, let's say 2 million. It means they save 2 million euros by using this new protocol for the cryopreservation. And this was mainly because of the samples produced on Friday. We waited then for the cryopreservation until Monday, 72 hours. And before, the Friday samples had to be thrown away about 45%, and in 2013, only 6%. So a tremendous improvement. And if you remember, I was already talking about this multicolor flow cytometric assay for the examination of cryopreserved sperm. And we looked what is important for the AI stations and also for us, if this has an effect on the fertility of the bulls. And it was there was a good relationship between the results of our assay and the non-return rate of the bulls. The bulls with higher non-return rate had more viable or intact sperm determined by our assay compared to the other samples. And what is important for the AI industry, not only to say, okay, we have a relationship, they would like to know, can I use the sample, yes or no? They need a decision tree. So and we noticed by using Foster analysis that the DNA integrity was the most important parameter for the decision. So this decision has been done by the computer, not by ourselves. by this Foster analysis. And if you see here, for example, samples with less than 6.4% DNA damage, they had and they also showed a higher value. And you see this means high fertile bulls, and these are low fertile bulls. And if they even had a higher DNA damage, more than 8%, all the bulls were low fertile. These are the dark bars. And if they were less than 8, between 6.4 and 8%, about 70% of the bulls were low fertile. But if they had a lower than 6.4% and lower than 4.6 in the DFI, they showed there were only 15% of the bulls were low fertile, and no low fertile if the calcium level was low in relation to the viability, and also a high percentage of low fertile bulls was if the percentage of this viable sperm was less than 70%. So coming to the conclusion, I hope that I could show you that flow cytometric assays provide accurate information about the quality of sperm. And the main advantage is the fast and objective determination of sperm for screening of sperm quality on AI stations. And this is not only good, important for practice, but also for science. And we have, by using this method, the flow cytometry, new options to improve the diagnosis of subfertile males and to optimize the sperm quality, for example, by optimizing the cryopreservation process. And then I would like to thank you for your attention, and I would like to ask, and I'm open for questions. Thank you.